So today I'm going to talk about the Hershey and Chase experiments. No, not that Hershey. This was a set of experiments done by these researchers who were looking to prove that DNA is the hereditary molecule, or the molecule that carries information between generations of cells. And we already talked about the fact that a lot of scientists insisted that the hereditary molecule was protein, which it is not. And this experiment really definitively proved that DNA is what is providing this information and transferring it between generations, and not protein. And this experiment works because it uses a much more simple system than the pathogenic bacteria and mouse system, which was used by the Griffith experiments. This system instead used these viruses, which are called bacteriophage. And the viruses look a little bit like this. They look a little bit like a spaceship. And they only infect bacteria. So these types of viruses cannot infect human cells. Of course, there are other types of viruses that can infect human cells, but not these. And these viruses are extremely simple. They only contain DNA and protein. Nothing else. No carbohydrates, no lipids. And the DNA and the protein are pretty much separate. So what they were trying to figure out in this experiment is, does the virus inject DNA, or does the virus inject protein into the host cell? Because we know that the hereditary information is injected into the bacterial host cell to make new viruses. And that's how you get the continuation, or the next generation of viruses in this type of virus. So here we see a bacterial host cell, and we have a virus particle. Again, viruses are not made up of cells, they're acellular. And so a virus can attach to the outside of the cell, but then it needs to transmit the hereditary information into the cell. And you'll see after a certain amount of time that new virus particles are going to appear inside of the host cell, whether that be a bacterium or, in other cases, a human cell or whatever. Information is transferred into the host cell, and it's used to make new virus particles. That's how viruses reproduce. And so we know that whichever type of molecule is injected is going to carry that hereditary information. So again, we can see here that after a while, regardless of whether or not the original virus particle that attached is there, we're going to see new viral particles inside of the cell, the host cell, and eventually that cell will either release them slowly or it will burst entirely and let them all out at the same time. The experimenters here are asking, or are proposing, that if protein is injected into the host cell, then protein must be the hereditary molecule. But if instead it is DNA that's injected, then DNA has to be the hereditary molecule. And the experimenters figure out a pretty ingenious way to differentiate which molecule is injected into the host cell. So the experimenters set up two different experiments, and you have to remember that the experiments were separate. They used everything the same except for the fact that DNA was radioactively labeled in one experiment, as we see on the left, and then protein was radioactively labeled on the right in a separate experiment. Now, the way that they were able to radioactively label the two molecules is that they used radioactive atoms of elements that were only found in either DNA or protein. So in DNA, or in the DNA experiment, they added radioactive phosphorus atoms into the broth so that when the new viruses were being made, their DNA would have radioactive phosphorus. And the phosphorus in DNA is, again, in that phosphate group. So if you recall, we have the sugar in the middle, the nitrogenous base on the right, and then the phosphate group. And that's what makes up a nucleotide. And the phosphate group would have that radioactive phosphorus. Then that DNA was going to be radioactive, and if it was injected into the host cell in step 2, as we see here, and used to make new viruses, then the experimenters would be able to track that because they would be able to see where the radioactivity was inside or outside of the cell. So again, this virus attaches to the cell, the bacterial cell. The virus has radioactive DNA, and the question is it injecting this DNA into the host cell to transfer instructions on making new viruses? Or on the other hand, is it proteins that are used as a hereditary material? Spoiler alert, it's not. And so one of the other great things about this experimental system is that the experimenters could wait and see if the hereditary information or the instructions on making viruses was transferred correctly, because then they could see if there were new virus particles being made inside of the cell, inside of the host cell, that is. And they could even allow that host cell to burst and then see if those viruses could infect new cells. So the other experiment on the right here is where they added not radioactive phosphorus, but instead radioactive sulfur atoms. And only 2 amino acids have sulfur, and it's only in their R group because every amino acid is the same in their central carbon, their carboxyl, their amino group, their hydrogen, but they're different in their R groups. But those 2 proteins, those 2 amino acids, that is, were enough. So those radioactive sulfur atoms were in the medium. They were incorporated whenever the amino acid was going to be made, and those amino acids were used in making the viral coat, or that exoskeleton spaceship-looking thing that we saw before. So again, that's one of the only other things that is in a virus. So excuse me for one moment to catch up to where we were before. We were talking about the viruses in the protein treatment attaching to the host cells, as we can see here. And so again, we have the host cell with the virus attaching. On the left, we have the DNA being radioactive. It's going to be injected into the host cell. On the right, we have the protein being radioactive. It's going to stay on the outside of the cell, and it just attaches. That's what actually attaches to the cell. But the DNA on the right is not radioactive, and the protein on the left is not radioactive. And so the experimenters were able to see whether or not the DNA or the radioactive material was injected into the host cell. So they did this by first taking a blender to dislodge those viruses from the host cells because they couldn't figure out another way. And then they separated the molecules, or the different molecules, by size and density, as we talked about in the last video. And they did this by centrifugation, and they didn't separate it too much, but they did separate out cells from non-cells. So all the cells ended up in the bottom of the tube and this is called the cell pellet. This happened in both the DNA-labeled experiment and the protein-labeled experiment. And at the top, there was a layer that did not contain cells. So it contained other stuff like just liquids, any fats that might have been in the media, or other things that weren't incorporated inside of the cell. And again, that's what's up here. So they were looking to see whether or not they saw radioactivity in the cell pellet, which would mean that whatever was radioactive was in the cell. And when they looked in each experiment, they saw radioactivity in different layers. In the DNA experiment, they saw radioactivity in the cell pellet. And that is, again, because the DNA was injected from the viruses into the host bacterial cell. And then we were making new viral particles. And those were infectious, and they did confirm this. In the other experiment, they did not see radioactivity in the cell pellet. They saw the radioactivity in the layer above that, meaning that the proteins did not enter into the cell. They remained outside of the cell. So the proteins, therefore, were not used as information to make new viruses. We still were able to make new viruses because the DNA was injected into the cell. It's just that in this experiment on the right, the DNA wasn't labeled with radioactive atoms. So again, in both experiments, there were new virus particles made. They were infectious. That was confirmed. But the difference is that the radioactively labeled atoms were in different places depending on whether we labeled DNA or we labeled protein. So again, on the right, they labeled protein, and so that was only in the coat of the virus, and it remained totally outside of the cell. There were new viral coats that were made inside of the host cell, but the materials at this point didn't include radioactive atoms, so those weren't incorporated. And so that's our experiment.